The cell wall of a bacterium encloses a riot of diverse chemical activity and molecules. To study the components of these cells, a researcher may want to break open the cells and purify the different molecules and structures. One useful method for breaking apart cells is sonication. In this method, a probe emits high-frequency sound waves which fracture the cells. The contents of the cells spill out into the medium, creating a complex suspension of molecules and fragments. The mixture includes unbroken cells, pieces of cell walls, cell membranes, and proteins such as enzymes. There are two common centrifugation strategies for separating the bacterial components. One is called differential centrifugation. In differential centrifugation, a tube is filled with a sonicated bacterial suspension and placed in a centrifuge. A second tube contains liquid for balancing the centrifuge during its spin. The first spin is usually set at a low rotation speed, producing a centrifugal force of about 15,000 times the force of gravity. During this short, low-speed spin, the largest and densest materials sediment first, forming a pellet at the bottom of the tube. The pellet contains primarily unbroken cells, flagella, and cell walls. The remaining liquid, called the supernatant, contains smaller and less dense structures. The components of the supernatant can be separated further by a second spin at a high speed, this time providing a force of 100,000 to 150,000 times the force of gravity for 60 minutes. Again, the materials with the greatest size and density move fastest to the bottom of the tube. Ribosomes and membranes sediment under these conditions. Soluble enzymes do not. Instead, they remain in the supernatant. Although differential centrifugation can quickly fractionate a bacterial suspension, the fractions are not pure. Another method, buoyant density centrifugation, provides greater purity. The suspension is centrifuged through a density gradient to separate the components. The tube in this example contains a sucrose solution with a high density at the bottom and a lower density at the top. The cell suspension is added to the top of the sucrose gradient. During centrifugation, the densest components move fastest down the tube. If a component reaches a sucrose density equal to its own density, it travels no further. The bands closest to the bottom contain components with high buoyant densities. Those components closer to the top of the tube